In my last video, already I have mentioned that the major complex biomolecules present in living cells are carbohydrates, protein, nucleic acid, and lipid. Among these, carbohydrate, protein, and nucleic acid, they are polymers and they are biomacromolecules, whereas lipid is biomicromolecule and it is not polymer, it is only composed of fatty acid and glycerol. Carbohydrates are composed of carbohydrates or polysaccharides are composed of monosaccharides. Protein is composed of amino acids. Nucleic acid is composed of nucleotides. Today in this video, I am going to discuss about carbohydrates in detail. Kindly stay with me till end of this video. Hello everyone, welcome to my channel Dr. TAPATI'S presentation. Today, topic of the presentation is carbohydrates. Carbohydrates are also referred to as saccharides or sugar. Probably these are the most abundant organic molecules in nature, and these are one of the essential constituents of living cells. These are mainly composed of carbon, hydrogen and oxygen, mostly in the ratio of 1:2:1. Few carbohydrates contain nitrogen and sulfur. Literal meaning of the name carbohydrate is hydrates of carbon or watered carbon. Therefore, the general formula of most of the carbohydrate is CXH2OY. However, there are few exceptions, like some of the genuine carbohydrates are there. They don't have the general formula of CXH2OY. Examples are rhamnohexose, which is with the formula C6H12O5, deoxyribose with formula C5H10 and O4. Therefore, carbohydrates cannot be always considered as hydrates of carbon. Another exception is that there are several compounds which also appear as hydrates of carbon, that means with the formula CSH2OY, but these are not carbohydrates. Examples are acetic acid with formula C2H4O2, that means it is also satisfying the formula, it is also the formula with CXH2OY. Similarly, lactic acid with formula C3H6O3, but these two compounds are not carbohydrates. Let's know functions of carbohydrates. Carbohydrate is the most abundant dietary source of energy for all organisms. It supplies 4 calories per gram energy. It is the precursor for many organic compounds like fat, amino acids. As glycoproteins and glycolipids, it participates in the structure of cell membrane and cellular functions like cell growth, adhesion, and fertilization. These compounds are structural components of many organisms, including fiber of plants, which is composed of cellulose, exoskeleton of some insects, which is composed of chitin, and the cell wall of microorganisms, means peptidoglycan. It is also serving as a storage form of energy as a glycogen in case of animal, and in case of plant it is starch to meet the immediate or short-term energy demands of the body. Carbohydrates are mainly classified into three groups: monosaccharides, oligosaccharides, and polysaccharides. Based on the functional group, monosaccharides are of two types: aldoses and ketosis. Aldoses means aldehyde group containing, ketosis means keto group containing. Based on number of carbon atom, they are either triose, tetrose, pentose, hexose, or heptose. Oligosaccharides, either disaccharide, trisaccharide, or tetrasaccharides. They are composed of mainly 2 to 10 monosaccharides. Polysaccharides means they are composed of more than 10 monosaccharides. They can be categorized into two groups: homopolysaccharides and heteropolysaccharides. Mono means one. Monosaccharides are the simplest group of carbohydrates, and these are also known as simple sugar. Mostly they have the general formula of CnH2ON. That means carbon is to hydrogen is to O ratio is 1:2:1. They cannot be further hydrolyzed by any enzyme. These are water soluble and sweet to taste. Already I told you, depending on the functional group, they are either aldoses or ketoses. Aldoses means aldehyde group containing. Examples are glucose and glyceraldehyde. Ketoses means keto group containing. Examples are fructose and dihydroxyacetone. Based on number of carbon atom, either it is C3 carbon containing means triose, then C4 carbon containing tetrose, C5 carbon containing pentose, C6 carbon containing hexose, and C7 carbon containing heptose. Glyceraldehyde, erythrose, ribose, glucose, and glucoheptose, these are examples of aldoses, whereas dihydroxyacetone, erythrulose, ribulose, fructose, and pseudoheptidose, these are the examples of ketoses. D-glucose, D-fructose, D-ribose, and deoxyribose, these are most important monosaccharides. D-glucose with formula C6H12O6. Fructose also same formula C6H12O6, but glucose is aldose because CHO group present, whereas fructose is ketose because it contains keto group. Ribose and deoxyribose both are aldehyde group containing, so these are aldoses. If you check the formulas of the glucose, fructose, and ribose, they are with the same formula, general formula of carbohydrate, that is CnH2ON. So C5H10O5, that is also CnH2ON, and D-fructose as well as the glucose with the formula C6H12O6, it is also with the general formula of CnH2ON. But if you consider the formula of deoxyribose, it is C5H10O4. So it is not using the general formula CnH2ON. Already I told you that carbohydrates cannot be always considered as hydrates of carbon. Deoxyribose is the proof for that. It is not using the same formula, general formula of the carbohydrate with CnH2ON. However, it is carbohydrate. Enantiomers are special type of stereoisomers. Those are non-superimposed mirror images of each other. The D and L isomers of monosaccharides are mirror images of each other. Those are known as enantiomers. The spatial orientation of OH group on the carbon atom, in case of glucose it is C5 carbon atom, that is adjacent to the terminal CH2OH group, determines whether the sugar is D or L isomer. Or you can consider that in the Fischer projection of the molecule, it is based on the position of OH on the chiral carbon. So here also it is C5 chiral carbon farthest from the carbonyl group. So C5 is the chiral carbon which is farthest from CHO group when OH group is on the right side of C5 carbon. It is known as the D series monosaccharide. Whereas if OH group is on the left side of the C5 carbon, especially in case of glucose, it is known as the L series of monosaccharide. So in this case, it is L-glucose. It is observed that naturally occurring monosaccharides in the mammalian tissues are mostly of D configuration. That means D-glucose is most predominant in nature because of the fact that the enzymes present in cells is specific to metabolize D series of monosaccharides. An aldehyde or ketone reacts with alcohol to produce hemiacetal or hemiketals respectively. Similarly, the hydroxyl group of monosaccharides react with its own aldehyde or keto-functional groups to form hemiacetal and hemiketal. The aldehyde group of glucose at C1 reacts with alcohol group at C5 carbon to form two types of cyclic hemiacetals, namely alpha D-glucose and beta D-glucose. In case of alpha anomer, the OH group held by the anomeric carbon, that is C1, is on the same side of the CH2OH group of sugaring, whereas in case of beta, OH group attached with the anomeric carbon is opposite side of the CH2OH group. These are the alpha and beta anomers of glucose in Fischer projection and Howard projection. Howard projection formula are depicted by a 6-membered ring pyranose and a 5-membered ring furanose. Pyranose is based on pyran structure and furanose is based on furan structure. Here I have given cyclic forms of glucose and fructose. First one is alpha D glucopyranose, which is 6-membered ring, and alpha D glucofuranose with 5-membered ring. And beta D fructopyranose is 6-membered ring, and beta D fructofuranose is with 5-membered ring. Mutarotation is defined as the change in the specific optical rotation due to the interconversion of alpha and beta form of D-glucose to an equilibrium mixture. The alpha and beta anomers of glucose have different optical rotations. Freshly prepared aqueous solution of glucose is in alpha D glucose form, and its optical rotation is plus 112.2 degree, which gradually changes and attains an equilibrium with optical rotation value plus 52.7 degree. Optical rotation of beta glucose is plus 18.7 degree. In the equilibrium mixture, 63% beta anomer present, then along with that 36% alpha anomer and 1% open chain form. In aqueous solution, the beta form is predominant, that means it is with 63%. It is predominant due to the fact that it is more stable as compared to alpha configuration. So beta D glucopyranose is more stable as compared to alpha D glucopyranose, and therefore in the equilibrium mixture, total 63% beta anomer present in the aqueous solution of glucose. In my last slide, I mentioned that the change in specific optical rotation due to interconversion of alpha and beta forms of D-glucose to an equilibrium mixture is the reason behind mutarotation of glucose. But in case of fructose, change in the specific optical rotation is due to the interconversion of pyranose and furanose structure. Crystalline beta D fructopyranose has an initial specific rotation of minus 133.5 degree, and it undergoes rapid mutarotation to minus 92 degree in equilibrium mixture. The equilibrated solution contains about 76% of beta pyranose and 20% of beta furanose form, 4% of the alpha furanose form, and very negligible amount of the open chain form. Second type of carbohydrate is known as the oligosaccharide. Oligo means short. That means these are short chain carbohydrates with 2 to 10 monosaccharides. Disaccharide, trisaccharide, and tetrasaccharides are composed of 2, 3, and 4 monosaccharides. Among various oligosaccharides, disaccharides are the most common. From the name, you can understand that di means two. So these are composed of two monosaccharides. Monosaccharides, either similar monosaccharides or dissimilar monosaccharides. They are attached together by glycosidic linkage, and the name of the glycosidic linkage is based on the linkages between carbon atoms and the status of the anomeric carbon, either it is alpha or beta. As for example, maltose is disaccharide which is composed of two molecules of alpha D glucose. So alpha D glucose, these are monosaccharides. this is monosaccharide. So two monosaccharides, two alpha D glucose attached through alpha 1,4 glycoside linkage. So in this case, bond between two monosaccharide, that is alpha 1,4 glycosidic linkage. And why alpha 1,4? Because it is anomeric carbon is alpha and it is C1, and which is attached with the second carbon alpha D glucose at C4 carbon atom. So 1,4 1,4 alpha 1,4 glycosidic linkage. Similarly, if you consider about the cellobiose, it is producing beta 1,4 glycosidic bond because uh, beta D glucose, two molecules of beta D glucose attached through 1,4 linkage, beta 1,4 linkage, therefore this name. And the name of the cellobiose you can consider in this way: beta D glucosyl 1,4 beta D glucose. So maltose and cellobiose both are composed of glucose, two molecules of glucose, but in case of maltose it is alpha D glucose, but in case of cellobiose it is composed of two molecules of beta D glucose. And in between their glycosidic bonds names are alpha 1,4 glycosidic bond, but if you consider about cellobiose it is beta 1,4 glycosidic bond. Sucrose and lactose are also disaccharides. Sucrose is composed of alpha D glucose and beta D fructose. Alpha D glucose C1 carbon is making glycosidic linkage with C2 carbon of beta D fructose. Therefore, sucrose is also known as alpha D glucopyranosyl 1,2 beta D fructofuranose. So in this case, glucose is in pyranose structure, whereas fructose is in furanose structure. If you consider about the lactose, it is composed of beta D galactose and glucose either in alpha or beta configuration, uh, and glycosidic linkage present in between C1 carbon of beta D galactose and C4 carbon of D glucose. So lactose is also known as the beta D galactopyranosyl 1,4-D glucose. Whereas lactulose is composed of galactose and fructose, not glucose. This is a synthetic disaccharide, neither digested nor absorbed in the intestine, but it is useful for the treatment of hepatic encephalopathy. Uh, Lactose can be isomerized to lactulose. Disaccharides are either reducing sugars or non-reducing sugars. Reducing sugar means with free aldehyde or keto group of anomeric carbon. Example is maltose and lactose. If you consider about maltose, maltose means it is composed of two molecules of alpha D glucose, and in that case C1 carbon of first alpha D glucose is attached to C4 carbon of second glucose. In that case they are making glycosidic linkage, but C1 or anomeric carbon of second glucose is attached with aldehyde or keto group. So it is so maltose is with free aldehyde or keto group. Therefore, it is reducing sugar, and it can reduce Fellings and Tollens reagent. Whereas if you consider about the non-reducing sugar, it is without free aldehyde or keto group of anomeric carbon. Examples are sucrose and trehalose. If you consider about sucrose, sucrose means it is composed of alpha D glucose and beta D fructose. In that case, C1 carbon, which is anomeric carbon of alpha D glucose, is attached with C2 carbon of fructose, which is also anomeric carbon. So both of the anomeric carbons are attached through alpha glycosidic linkage, and therefore there is no free aldehyde or keto group at the anomeric carbon. Hence, sucrose is non-reducing sugar, while all monosaccharides are reducing sugar because they are with free aldehyde or keto group of anomeric carbon. There is no glycosidic linkage. In a polysaccharide, the individual monosaccharides are linked by a glycosidic linkage, and this bond is formed by the dehydration, or you can consider one molecule of water is removed So one molecule of water is removed so that glycoside linkage is produced in between two monomers. I have given here maltose as the disaccharide where 2 alpha D glucose present in between 2 alpha D glucose alpha glycosidic linkage present. Whereas I have given another disaccharide, that is cellobiose, which is composed of two molecules of beta D glucose. Therefore, in between these two glucose molecules, beta glycosidic linkage is present. So maltose and cellobiose are disaccharides. So similar way for the polysaccharide, n number of glucose present in case of starch if you consider. So in that case, in starch molecule, alpha glycosidic bond present because it is composed of repeating units of alpha D glucose. Whereas if you consider about the cellulose, it is composed of repeating units of beta D glucose and therefore beta glycosidic bond present. So you can consider glycosidic linkage two types, alpha and beta, and sometimes you will find in this case you are getting that 1,4 alpha 1,4 linkage present, but somewhere you will find alpha 1,6 bond also present, especially if you consider about the glycogen and starch molecule. Along with the alpha 1,4 linkage, alpha 1,6 linkage also present. Third type of carbohydrate is polysaccharide. Poly means many. That means polysaccharides are composed of many monosaccharides, mainly more than 10 monosaccharides present in a single polysaccharide. Suppose this is one polysaccharide with in that polysaccharide more than 10 monosaccharide present. So these beads are monosaccharides. So monosaccharides are building blocks of polysaccharides. So in this picture, in this graph, in this polysaccharide, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. So total 11 monosaccharides present. So these are actually monosaccharides. The beads are monosaccharides, and in between two monosaccharide, how many glycosidic linkage present? So this is 1, 2, 3, 4, 5, 6, 7, 8, 9 and 10. So total 10 glycosidic linkages present in between monosaccharides in this case. If you suppose 11 monosaccharides present, so in between 11 monosaccharides total 10 glycosidic linkages present in a polysaccharide. Now polysaccharides are two types, homopolysaccharides and heteropolysaccharides. Homopolysaccharides are composed of a single type of monosaccharide. That means on hydrolysis by acid base or carbohydrate enzyme, homopolysaccharides eat only a single type of monosaccharide. Examples are starch, glycogen, cellulose, inulin and chitin. Starch and glycogen are composed of monosaccharides, that is alpha D glucose. So building block of starch and glycogen, that is monosaccharide alpha D glucose. And in these two polysaccharides, alpha 1,4 and alpha 1,6 glycosidic linkage is present, where alpha 1,6 glycosidic linkage is known as the branching point. Difference between starch and glycogen is that although they are composed of the similar type of monosaccharides and similar type of glycosidic linkage is present in between two glucose molecules, but branching points are more in case of glycogen. That means 1,6 glycosidic bond, number of 1,6 glycosidic bond would be more in case of glycogen as compared to starch. Cellulose is also composed of glucose, but it is composed of beta D glucose. So building block of cellulose is beta D glucose, where beta 1,4 glycosidic bond present, and there is no 1,6 glycosidic bond. That means branching point is absent in case of cellulose. Inulin is composed of fructose and beta 2,1 glycosidic bond present in between fructose molecules. Occasionally, it has a glucose residue at the end linked by an alpha D glucopyroxyl bond. Chitin, which is present in exoskeletons of arthropods and fungal cell wall, it is composed of N-acetyl D-glucosamine, which is an amino sugar, and beta 1,4 glycosidic bonds present in between in between two N-acetyl D-glucosamine molecules. Starch is the carbohydrate, that means polysaccharide, and it is a reserved polysaccharide or reserved carbohydrate present in plant, and which is most important dietary source for higher animals including human. High content of starch is found in cereals, roots, tubers, vegetables, etc. And glycogen is the carbohydrate, reserved carbohydrate for human, animals, hence often referred to as the animal starch. And it is present in high concentration in liver, followed by muscle, brain, etc. And it is also found in plants, but do not possess chlorophyll. That means in case of yeast and fungi, glycogen present. Whereas heteropolysaccharides are composed of a mixture of monosaccharides or their derivatives. That means on hydrolysis of heteropolysaccharides yield mixture of monosaccharides or their derivatives. Mucopolysaccharides or glycosaminoglycans, that is a GAG, are most popular heteroglycans or heteropolysaccharides, which are made up of repeating units of sugar derivatives, namely amino sugars and uronic acids. Examples of glycosaminoglycans are hyaluronic acid, chondroitin sulfate, heparin, and dermatan sulfate and keratan sulfate. Agarose and pectins are also examples of heteropolysaccharides. Hyaluronic acid is composed of alternating units of D-glucuronic acid plus N-acetyl D-glucosamine, which are attached through beta 1,3 glycosidic bond and beta 1,4 glycosidic bonds. Whereas chondroitin sulfate is composed of D-glucuronic acid plus N-acetyl D-galactosamine 4 sulfate, and glycoside linkages are beta 1,3 glycoside bond and beta 1,4 bonds. Heparin means it is composed of repeating units of N-sulfo-D-glucosamine 6 sulfate plus glucuronate 2 sulfate. Alpha 1,4 glycosidic bond present in between these two monosaccharides. Dermatan sulfate is composed of L-uronic acid plus N-acetyl galactosamine 4 sulfate. Beta 1,3 glycosidic bond present in between these two monosaccharides. Keratin sulfate means it is composed of repeating units of D-galactosamine plus N-acetyl glucosamine 6 sulfate. Beta 1,4 glycosidic linkage present in between these two monosaccharides. Agarose is composed of galactose and anhydrogalactose and alternate alpha 1,3 and beta 1,4 glycosidic bonds present. In case of pectin, it is composed of galacturonate and rhamnose. In your NCERT book, it is mentioned that in a polysaccharide, as for example glycogen, the right end is called the reducing end and the left end is called the non-reducing end. So in this picture, this is a figure of glycogen. So in that case, this part, that means right end, is known as the reducing end, whereas left part is known as the non-reducing end. Now why right side is right end is reducing end? Because of the fact that in this case C1 carbon is free. That means CHO group attached with C1 carbon. C1 is the anomeric carbon. So CHO group is free here. It is not involved in the glycosidic linkage. So due to the presence of free CHO group, it has reducing power. But if you consider about the left end, in that case C1 carbon is involved in the glycosidic linkage because it is making alpha 1,4 glycosidic linkage with the second glucose molecule. So suppose this is one glucose, and then with the next glucose there will be bond with C1 and C4 carbon atoms. Now, if you consider this is one glycogen molecule, so in that case, this polysaccharide is composed of alpha D glucose, and in between alpha D glucose, alpha 1,4 glycosidic linkage present. Whereas if you consider here another glycosidic linkage present, which is alpha 1,6. This one is alpha 1,6 glycosidic linkage. So both alpha 1,4 and alpha 1,6 glycosidic linkage is present in case of this is alpha 1,6. This point is alpha 1,6 glycosidic linkage. So alpha 1,6 glycosidic linkage means these are branching points. Alpha 1,6 means branching points. Alpha 1,6. These are alpha 1,6 glycosidic linkages. But in this case, suppose these are monomers of glucose, monomers of glycogen. So these are glucose molecules. In between two glucose molecule, alpha 1,4 glycosidic linkage present. Alpha 1,4 glycosidic linkage. So this is the diagrammatic representation of a portion of glycogen. Similar type of structure you will find in case of starch molecule. A very important field in which enzymes have proved to be of great value over the last few decades is the starch industry. Starch is a major energy compound of many economically important crops such as wheat, rice, maize, tapioca, potato, etc. Besides, as a versatile raw material, it has been used in a variety of industrial applications and has a great impact on the world economics. Starch is usually composed of two types of molecules, namely amylose and amylopectin. They both consist of polymers of alpha D glucose units in their chair conformation. That means basic unit of amylose and amylopectin is alpha D glucose. Amylose is unbranched single chain polymer of about 500 to 2000 glucose subunits with only alpha 1,4 glycosidic bonds. Whereas in case of amylopectin, it is actually branched both 1,4 glycosidic bonds and alpha 1,6 glycosidic linkages present in amylopectin. The degree of branching is approximately 1 per 25 glucose units in the unbranched segments of amylopectin. Amylose exists mainly in amorphous form, whereas amylopectin forms the crystalline component. So these are the basic differences between amylose and amylopectin. First picture is showing amylose, that means it is unbranched single chain polymer of glucose, where you can see alpha 1,4 glycosidic linkage present. Whereas if you see second picture, this is actually amylopectin, where alpha 1,4 link is present, as well as alpha 1 alpha 1,6 glycosidic linkage present. Glycogen is another closely related compound functioning as the glucose storage in animal cells, which has one branching per 12 glucose units. That means it has similarity with amylopectin. It is actually branched both alpha 1,4 glycosidic bonds and alpha 1,6 glycosidic bonds present. And already I told you it has a one branching per 12 glucose subunits. So more branching present in glycogen as compared to amylopectin, and it is basically present in animal cell as storage compound. In this figure also it is clearly shown that in case of amylose only alpha 1,4 glycosidic linkage is present. In case of amylopectin, both alpha-1,4 glycosidic linkages as well as alpha-1,6 glycosidic linkages present. That means branching point present in case of amylopectin. In the full view of amylopectin, you can see that there are mainly three branching points. This is one branching point, then another branching point is here, and then third branching point present here. Branching point means mainly that is alpha-1,6 glycosidic linkages. Whereas if you consider about the amylose, there is no alpha-1,6 glycosidic linkage. That means there is no branching point in case of amylose. In this figure, you can see this is a reducing sugar, reducing end of the amylose, and in this in case of amylopectin, this point, the black circle, is representing the reducing end of amylopectin. So both the cases, amylose and amylopectin, there will be presence of reducing end. If you think this video is helpful for you, kindly like, share and subscribe this channel.